Okay, so I'd like to welcome our next speaker, Dr. Ed Boyden. So he is the Y. Eva Tan Professor in Neurotechnology at MIT, leader of the Synthetic Neurobiology Group at MIT's Media Lab, co-director of Neurobiological Engineering, and he's been an investigator at the McGovern Institute since 2006. He conducted his PhD work at Stanford, during which he invented and developed a groundbreaking technology, optogenetics, which has revolutionized neuroscience, as Bob mentioned earlier. He's received many awards and accolades and was selected as, for a prestigious HHMI investigator position recently. Ed is developing tools that image and control the brain and applying them to solve entire brain circuits. This ambitious goal is exemplified in his continuously innovative and highly collaborative research and creativity. And he has the intriguing title, From Structure to Function, Optical Tools for Analyzing the Complexity of the Brain. Dr. Ed Boyden.
Thank you. Well, it's really a great honor to be here today and remembering Pat McGovern, and who, along with, of course, Lori, co-founded this fantastic institute. It's been transformative to our group to be part of it and to be collaborating with such amazing people, because in the end it is all about the people, and to work with great colleagues, collaborators, and friends. So I direct a group here at the McGovern and also at the MIT Media Lab, which is focused on trying to figure out how to link the underlying building blocks of the brain to the emergent dynamics and functions. And a lot of our work has been technological in nature, but we're also finding ourselves increasingly tackling scientific questions as well, as the tools get better and better. At this point, I would say that the overarching question that gets me out of bed in the morning is this question of linking structure and function. Could we make maps of the brain, the wiring, but not just the wires, the molecules along the wires and at the connections between brain cells, and measure behavior and dynamics and perturb behavior and dynamics in a way that lets us connect these levels of description? My dream is someday that we could, let's say, going from bottom to top here, watch neural activity throughout neural circuits with enough precision, spatial and temporal, that when we perturb them, we can, the middle controlling topic there, be able to test in a causal way what patterns are important. And then finally, the top part there in the blue, map the molecules and wires and synthesize these data sets into models of how brain circuits operate. My dream is that these models could be someday biologically realistic and human understandable. Of course, there's no guarantee of the latter, but we are optimists in science. Up till now, we've been focusing on tools that really obey two sort of general design principles. And it helps to sort of discuss why the brain is so complicated, and there's many, many reasons. But one of the issues, of course, is the tremendous differing in spatial scales that we deal with. So brain cells are enormous. In the human brain, they're centimeters in spatial extent, or if you count going down the spinal cord, a meter in length. And yet if you care about the wires in the brain, axons and dendrites, or the connections like synapses, those are nanoscale in dimension and chock full of nanoscale building blocks, biomolecules, of which there are somewhere between tens of thousands and millions. We actually don't know the exact number. So how can you see and ideally control biological building blocks and functions across these incredible spatial scales? This represents a real challenge, and a lot of the older tools that were developed for smaller ranges of scales in biology don't generalize well to the brain. And the other issue is time. So similarly to the spatial scales, the temporal scales that we deal with are really daunting. You know, if you care about learning or neurological disease or development or aging, these are time scales of years or decades we're talking about. And yet the quantum building blocks of neural computations are electrical pulses within brain cells and chemical exchanges between brain cells that last 1,000th of a second. So one of our day jobs in our group is building tools that help neuroscientists cross these spatial and temporal dimensions. So today I'll tell you 3 short stories that end with a view towards the future about how we hope to develop and refine the approach to neuroscience that really allows us to cross these spatial and temporal scales in ideally an integrative fashion. These tools should work together well and be used together to coherently answer scientific questions. So let's start with mapping. How do you map the molecules throughout brain cells and the brain cells throughout entire circuits? So around the time we began this project, there were many people working on ways to image brain circuits. For example, electron microscopy gives you incredible resolution, but struggles to give you multiplex molecular information from inside the brain. And many people have been working on super resolution microscopes. In fact, a few of the inventors even won the Nobel Prize in chemistry a couple years back. But these tend to be slow and difficult to apply to extended 3D objects. So in our group, starting with two then grad students, Fei Chen and Paul Tilburg, and now about half our group works on this, decided, what if we do the opposite of what people have been doing for hundreds of years? Rather than zoom in onto a piece of a cell or piece of a tissue, let's do the opposite and blow it up. Then you could use really inexpensive optics, scalable optics, to do 3D imaging with nanoscale precision, because if we blow up the brain in an even fashion, that takes care of the magnification for you. So after thinking about many ways of doing this, we converged upon the following idea. What if you could actually take a swellable polymer, like the stuff you find in baby diapers, sodium polyacrylate, and weave a spiderweb-like mesh so dense and so evenly around and between the biomolecules of the cells of the brain that you could actually pull the biomolecules apart, so that a brain cell like the one on the left side of this cartoon, shown in gold, would turn into something like the one on the right, a constellation of biomolecules hovering in space, like stars in a constellation in the sky, but anchored to the polymer and expanded evenly from each other. Now to do that, we had to invent several new kinds of chemistries, or borrow chemistries from the field of chemical engineering. One of the things we had to borrow were anchoring molecules. So in this cartoon, the brownish blobs are proteins, and we have now developed anchoring molecules, or found ones that are commercially available, that will bind to proteins, DNA, RNA, and in our own group, we have unpublished work where we're working on lipids, and also with Laura Kiesling groups here at MIT on sugars. So the purplish blob here is an anchoring molecule that binds to the protein and equips it with a handle. Next, we had to synthesize a polymer mesh throughout the specimen, and to do that, we can soak the specimen in a solution of monomers, shown as the little white spheres, and they self-assemble into long polymeric chains, shown as assemblages of the white spheres. And when these growing chains encounter a handle, they'll form a covalent bond. So if you think about it, this white polymer is a swellable polymer, so just as the stuff in baby diapers, if you add water, it should swell and provide force to the handles, which will then pull the biomolecules apart. But there's one last thing we had to figure out, and that's how to soften the tissue, because the molecules are very happy where they are. And so we use detergents, enzymes, and or heat to loosen up, or sometimes even destroy, if we don't care about it, biomolecules. And then the water will cause the sodium polyacrylate, the baby diaper material, to swell, but now the biomolecules will come along for the ride. So we first reported the discovery that we could evenly swell a biological specimen back in 2015. The swelling is not perfect. There's a few percent of distortion over length scales of tens to hundreds of microns, but for the vast majority of biological and medical questions, that's okay. And we are working to make it even more even for the subset of questions that require more precision. In panel B is a piece of the mouse brain, and in panel C on the right is the same piece of mouse brain tissue about a day and a half later, when we have blown it up by about 100 times in volume, about 4.5 times in each direction. And in the upper left is a cartoon of the dense polymer mesh. We know from previous literature from chemical engineering, going back many decades, that the spacing between polymer threads is really tiny, maybe as low as 1 to 2 nanometers, around the size of a biomolecule, or even smaller. So when the polymer mesh swells, like in the schematic in the lower left, we can anticipate, with good reason, that the design of our chemistry will allow us to get precision down to some number of nanometers. And we and many others now have validated the resolution down to around 20 nanometers. Below that, the jury's still out, and we're working to refine it further, and so stay tuned. So here's a little movie of a piece of mouse brain tissue that's been polymerized, and now we're adding water to expand it. The movie's sped up by about a factor of 50, but I hope you can see that it's growing right before your very eyes. It's a pretty fast process. The whole process takes about an hour, let's say, and it's pretty easy to learn. We've now taught well over 300 research groups how to do it, and I'll show you later it's being applied to all sorts of stuff. Now, as I mentioned, the expansion is not perfectly even. We've done a lot of validation to try to see how accurate it is. The polymer expansion process is pretty good because of its design that I told you about, but also because we've validated it in a wide variety of tissues for a wide variety of species. And so what we do find by comparing post-expansion images to pre-expansion images taken with classical super-resolution methods, they're slow and expensive, but they work well over small volumes, is that we actually are getting pretty good low distortion levels. So here's an example of actually some data from our very first paper on it. The left column shows us zooming in into a Thai-1 YFP mouse that Guoping and colleagues made many years ago. And on the right is the same zoom in after we expand. And I hope what you can see, the color codes explained on the left, we have anti-bassoon in blue, a presynaptic protein, and anti-Homer in magenta, a postsynaptic protein. As we zoom in iteratively into the white squares, we eventually hit the resolution limit of the microscope, because light has a finite size or wavelength that you can't see much smaller than. But the right-hand side, we can go all the way into even parts of synapses. So here is blue and magenta, the presynaptic and postsynaptic densities that are on either side of the synaptic cleft. And in the upper right, if we measure the distance between the centroids of these two regions, we get the same answer that using storm microscopy, Zhao Zhuang's group found many years ago, of the distance between these. But now we can get this kind of information on regular old confocal microscopes, which are practically ubiquitous in neuroscience. Now, one fun thing about this process, as a byproduct of the expansion process, is that you end up with something that's like 99% water, because you've soaked it in water and the water has drifted in via osmosis, and it swells it, so you get something that, as you can see on the left-hand side, ends up pretty much perfectly clear. So in panel A is a piece of the mouse brain in the upper left, and in the lower left in panel B is the same piece of mouse brain tissue, and it's completely transparent and refractive index homogenized. Now, why is that interesting? Well, as we found, and also Oswasi, when he joined our team, and many other people helped us in the study, if you expand something and it becomes transparent, you can then shine light from the side, let's say, and take a picture from the top, so-called light sheet imaging. And then you can go much, much faster than before. In fact, working with Eric Betzig, one of the winners of the Nobel Prize for his work on super resolution, and now a convert to expansion microscopy, I'm happy to say, we actually collaborated to use one of his light sheet microscopes, a lattice light sheet microscope, to image expanded brain tissue. So in a collaboration triply spearheaded by Rei Gao, Sho Asano, and Goku Opariula, working across many of our groups shown in the lower right, we expanded brains, labeled them with antibodies that bore fluorophores, and then by using one of these clever lattice light sheet microscopes that Eric's group invented, we found that we could image almost 1,000 times faster than a comparable resolution previous super resolution method. And it's only a matter of engineering, we think, to go much, much faster. So we're talking about many, many orders of magnitude improvement in our ability to rapidly scan brain circuits. So shown here on the upper right, let's say, are mitochondria in purple and lysosomes in yellow, stained with antibodies. Here we are imaging the thickness of the mouse cortex with layer 1 on the right and layer 6 on the left, same color code as shown before, and the insets are zoomed in the bottom. So rather than go through all the panels one by one, I'll show you just a movie here where in yellow are those famous Thai-1 YFP mice glowing with yellow fluorescent protein, and in blue and magenta is the same color code as before, showing pre- and postsynaptic sides of synapses. That's kind of a long movie, so I think I'm going to skip to a part in the middle where you kind of zoom in a bit, and you can start to really see the ability to look at individual synapses, but not lose sight of the overall architecture of the neuron. And that's really, I think, at the soul of this question that I posed at the beginning. How do you cross spatial scales, which are bearing this tension, where you don't want to lose sight of the nanoscale building blocks like synapses, but you sure also don't want to lose sight of the architecture of neural circuitry as well. Eric being at Geneali Research Campus, we of course had to do some work on Drosophila. Whoops. Let's see if this movie works. This is one of my favorite movies because we actually will fly through the fly brain. Here we have dopamine neurons expressing fluorophores. That's what you see shown here in the outlines of neurons. Of course, dopamine is so important for so many things. But if you could image all the neurons in an entire brain and link those neural architectures to the connections and the molecules that are in those neurons, we're very excited now that whole brain imaging of circuitry and building blocks of circuitry might now be within grasp. And on that note, one of the most immediate applications of this kind of work is to look at the wiring of the brain. This is some work that we published a couple years ago, collaborating with Dawen Kai, one of the recent pioneers of rainbow techniques when he was working with Jeff Lichtman and now in his own group at Michigan. And what you can see here in the lower panel is a piece of mouse hippocampus with rainbow, namely fluorophores genetically encoded that are virally expressed in random combinations. So the blue cell and the green cell express different fluorophores. Maybe some of the aqua and blue-green cells express combinations. And at the top middle panel, you can see what it looks like when you zoom in. Beautiful colors, but it's very hard to see individual axons, especially when they're close by. But in the upper right is what you get after you expand. So of course the image is much bigger, but we shrank it to fit on the slide. And I'll just use my mouse pointer to highlight this little green banana shape here. Before you expand, it's really hard to know what we're looking at. Here we are in the hilus of the mouse hippocampus. But after you expand, you can cleanly resolve individual axons, even in this very dense neuropil-rich arena. So we're very excited now about scaling up our color coding and also scaling up our expansion factors, as I'll show you in a second. Importantly, you can look at things other than proteins. So far, everything I've shown you is a protein. But you can also, and this is work that Fei Chen Oswasi, in collaboration with Arjun Raj, Alison Coate, and Arjun's group, and others, we worked on together. We developed anchors that bind to RNA. So now you can bind expressed genes at the RNA form, expand them, and then do fluorescent in situ hybridization afterwards. And just to focus on the right-hand side of the slide, we're looking here at classic dendritically localized RNAs like chem kinase 2, and now we can see them along dendrites or even in synaptic spines, which is very difficult to do in intact brain tissues. And this is, to my knowledge anyway, the first nanoscale resolution imaging of single RNA molecules deep in intact brain circuits. If you can expand RNAs from each other, maybe you can also use the room that you make around the molecules that have been decrowded to run interesting biochemistries. And so we just submitted a paper, which was sent out for review, where we adapted the George Church's lab's in situ sequencing chemistries to work in expanded brains. So their group had, to my knowledge, the only untargeted in situ sequencing method. You don't want to hybridize to a known target, you just want to sequence everything and find out what cells are expressing. Unlike other ways of sequencing, where you grind up the tissue and extract the RNA, they're basically running sequencing chemistries right inside cells. Now, when we tried it in tissue, it didn't work very well. So what we decided, this is work triply spearheaded by Shahar Ulon, Dan Goodwin, and Fei Chen, along with great contributions from Evan Doherty and George's group. If we anchored the RNAs and then expanded them away from each other, we could then amplify the resulting cDNAs after reverse transcription and then run sequencing. So when you run sequencing, you're basically copying the nucleic acid, and as you copy it, you add fluorescent nucleotides. And so what happens is, over many rounds of sequencing, you will get these individual gene expression products located at different points in the cell to blink out their identity. And that's exactly what we got. So in the lower right-hand panel, you can see 20 rounds of sequencing going down these columns, and each of these colors, blue, green, magenta, and red, correspond to letters of the genome. So what does that mean? Well, again, there are lots of ways of doing targeted analysis of transcripts in tissues. But our method is, to my knowledge, the first method of doing untargeted analysis of transcripts in intact tissues. So what we can do here is take a piece of brain, here we are in the mouse hippocampus again, expand it, and then look at the identity and location of expressed RNAs in the circuitry, even in the context of the shapes of the cells, the morphologies of the cells, and maybe someday soon the connections. So what does it mean to be untargeted? It means we don't have to have a hypothesis about what genes are expressed. So we can look at, for example, things like different transcription factors, which, of course, do their business in the nucleus, but you can find them hanging out in the dendrites, far away from the nucleus. Intron-containing transcripts. Neurons have one nucleus, but thousands of synapses. People have found evidence that neurons do splicing later after the mRNAs are shipped out of the nucleus, but here now we can actually pinpoint where they are. That's what's shown in magenta. And all sorts of other things as well, structural RNAs, and so on and so forth. So we're very excited now, if you put 2 and 2 together, that we could use color codes like I showed you earlier to get the maps of the wiring, and then untargeted in situ sequencing to get the maps of gene expression, and integrate these all into a cohesive strategy for mapping out the building blocks and wires in neural circuits. Now, to do it with very fine resolution, you might need to expand more than the 100x volumetric expansion that I showed you. And so when J.B. Chang was a postdoc in our group, we decided to try to magnify things bigger. How do you do that? Well, you can take a piece of brain and polymerize it and expand it like I showed you earlier, and then you can form a second polymer, shown in this cartoon in orange, in the space opened up by the first, and expand it again. So it's kind of like PCR, but for space. You get exponential amplification in space with a linear number of steps. And it works. So in the upper left here, just to pick one example, and I know it's a crowded slide, before I showed you Homer and bassoon, which we often use as just a ruler, and before you saw those two protein densities touching, after double expansion, which gives you a 20x linear expansion, there's now a gap between them. And sure enough, if you antibody stain with an antibody against glutamate receptors, which are of course there at the synaptic cleft, you can see them in between the pre and post-synaptic densities. And something similar holds for inhibitory synapses. Going back to brainbow, in the upper left you can see an unexpanded piece of brain tissue. In the upper right, after you expand once, so you can see many of the wires, but some things like spine necks are still hard to resolve. And then in the lower left is double expansion. And I hope you can see here that the image looks kind of punctate, kind of speckly. And so we really think now that we're getting down to nearly single molecule detection, although we have not yet proven that we have single molecule resolution. As I alluded to earlier, we still have to validate under the 20 nanometer length scale. Yongxin Zhao and Octavia Bucur, working between my group and several pathology groups, Astrid Wines, Duschit, Andy Beck, we got it working in human tissues. So this has now been applied to a wide variety of human tissues, including human brain tissue. And I think I'll end this part of the talk just by pointing out that many, many people have started to use expansion in many, many contexts. We had a great collaboration with Anne Graybiel, shown in the upper left, to look at striatal circuitry. It's been applied to bacteria in the upper left, human brain in the upper middle. Our group has been working with several other groups on C. elegans in the lower left, work spearheaded by J.U. in our group with the help of many others. And all the protocols are on our website, expansionmicroscopy.org, and we're happy to help teach usage of the tools and to explore how to improve them if you give us feedback as well. So that's the first story I want to tell you about. And in the last third of the time remaining, I really wanted to talk about live imaging and live control. So of course, expansion only works on preserved things. We do have some ideas of how to try to preserve some living functions in expanded things, but of course, everything's anchored in place. So diffusion won't work, and it's a really weird environment. So in terms of live control and imaging, of course, we've been working for many years on optogenetics, where we use light to control neurons that express a genetically encoded construct. Why light? Well, when Carl Deisroth and I started brainstorming about this, we decided just to go through all the laws of physics, magnetism, ultrasound, etc., and decided light would be great if you could pull it off because you could aim it at things, and also it's as fast as anything ever gets, so you could actually keep up with the high speed of activity of the brain many, many times over. And then you have to get light into the brain. As you've already seen in earlier talks, this is something that is not so hard to do if you use a device that conveys light into the brain instead of an electrode, which of course has been used for many, many years. So then the question becomes how to make brain cells sensitive to light. Well, it turns out the natural world has evolved a class of molecules that does just that. So over the last half century, many people have characterized light-driven ion pumps and channels that convert light into electrical gradients. So on the upper left is an image of some salty water near the edge of a body of water. And in the lower left is the crystal structure of a pigment that's found in a microbe that lives in such salty water. And this pigment doesn't just do nothing. It converts light into an ionic gradient by pumping protons from one side of the membrane to the other. That was called bacteriorhodopsin. Later, several groups found halorhodopsins, light-driven chloride pumps that do something similar, but for chloride. These are pumps that convert light into electrical gradients, so they're thought to be serving a photoenergetic role and to store energy in chemical form. And then around the turn of the millennium, light-driven ion channels were found. Being channels, of course, they don't store energy, but they can be used to tell single-celled algae where to go in bodies of water. So to make a long story short, what we found over the years, and this is work that's been led by many people in the group, including Brian Chow, Xue Han, Amy Chuang, Nathan Klepetke, and others, is that members of all three of these classes of light-driven ion pump and channel can be found that are safe, effective, and fast enough to work in delicate mammalian neurons. And this is a lot of serendipity. We didn't have to engineer these molecules almost at all. And amazingly, I forgot to mention this, but these molecules have a chemical cofactor called all-trans-retinol. And amazingly, we don't have to add that chemical. These proteins worked out of the box in intact mammalian brain circuits. So a lot of luck lined up here. Anyway, with light-driven proton pumps and light-driven chloride pumps, we found that we could either bring positive charge out of neurons or negative charge into neurons, which allow us to shut down neural activity. And with light-driven ion channels, which the earliest of which would let positive charge into brain cells, we could activate them. And more recently, people have found and made versions that also will let negative charge into cells. And these come in different colors. The colors shown here, green, orange, and blue, are the colors originally used to drive these. But in recent years, with Amy and Nathan's PhD theses, we've been trying to find molecules that are sensitive to red light, because red light, of course, goes deeper in the body than green light. This is a green laser pointer, but if I shine on my finger, you can't even see it. And if we had a red laser pointer, I could do the opposite, where you would see my finger glowing. So where's the frontier here? Well, we have a very close collaboration with Valentina Miliani's group to try to push these molecules to the point of single-cell resolution. Up till now, most groups have been using optogenetics to activate or silence populations of neurons, turning on and off a set in synchrony with each other. And that's great. Much we've learned about neurons by doing so, but wouldn't it be great to be able to activate neurons individually, since even two identical or very close in phenotype cells nearby to each other could have different neural codes. So Urs Shemesh in my group, and Dimitri Tanese and Valeria Zampini in Valentina's group, worked together to use Valentina's holographic microscope projection systems. They make interesting patterns of light like the one you see at the bottom here. Could you use these to do good single-cell resolution control? So to do that, we had to find a way to target the expression of these microbial opsins, as they're known, to just the cell bodies. This is not our idea. Many groups have done this before, like McLean Bolton's, Jawa Pons, and Frank Werblin's. But it solves a problem that is tricky, which is namely if you use microscopes that, like Valentina's, to aim light at a single cell, shown here in the dotted lines, you activate the stray cells nearby whose axons and dendrites happen to wander around the cell of interest. If you can target to the cell body, though, then aiming light at that cell body will be enough to clean up the background because the other axons and dendrites will be bare. So in our group, we started wondering, McLean and Jawa and Frank had targeted one of the older opsins called channelrhodopsin 2 to the cell body. What if we tried to actually do a dual search in genomic space as well as in engineering space to find a molecule that was an order of magnitude better? And it turns out that Nathan, when he was discovering a red light activator that we call Crimson many years ago, he also found a molecule that we called COCHR, which has 10 times bigger photocurrents than the channelrhodopsin 2 that was put into neurons by Carl Deisroth and myself many years ago. And if we'd known it was going to be a cool molecule, we would have called it something more interesting than COCHR, but we weren't that visionary. Anyway, or a postdoc in the group found a peptide from a kainate receptor that we fused to COCHR, and this is in the mouse cortex. You can see that the background fluorescence, indicated by GFP fused to the opsin, really gets cleaned up when we have this peptide from kainate receptor 2 attached to it. And so now when we do holographic stimulation using Valentina's holographic projector, we can actually then get activation of single cells without activating stray cells. So if we patch a cell and then scan around, with the regular COCHR, a third of the time we get stray activation, but with the soma-targeted version, we almost never get stray activation. And because COCHR is so powerful, we can use brief, bright pulses of light and get sub-millisecond jitter, or temporal error of the action potential. So that's the update I wanted to share with you on the optogenetic control front. And I just wanted to emphasize one thing, which was the enormous amount of serendipity that occurred. We were lucky to get molecules that were safe, fast, and effective, basically right out of the box. And they also were able to use the all-trans retinal that, for some reason, is found actually in the mammalian brain. But what about the opposite, in terms of imaging neural activity? There we're not so lucky. The natural world has not provided us, through evolution, molecules that will light up neurons when they're active, and so we have to work hard to make them. And so Kirill Pijakovic and Erica Jung, who are currently and former postdocs in the group, along with many collaborators shown in the lower right, we decided if nature's not going to evolve these for us, we're going to evolve these things ourselves in the laboratory. Now, directed evolution is a very hot topic, won the Nobel Prize in Chemistry recently, but doing it in an automated way would really help optimize these difficult to optimize molecules like fluorescent indicators of neural activity. And we chose to build voltage indicators because then we can really get very fine-scaled information in terms of the temporal and amplitude properties of synaptic events, action potentials, and subthreshold oscillations. So what you see here is basically a robotic scientist we're building. The top row just shows how we take a gene of interest. In this case, we started with Adam Cohen's quasar 2 molecule, a candidate fluorescent voltage indicator that works well in cultured cells, but is dim and poorly localized in vivo. And then the bottom part of the slide, you can see how we pick the cells. We select for cells that have mutants of quasar 2 that are bright, fast, and photostable and well localized in the membrane. And then we use a robotic arm to pick out the cells and therefore the mutants that have interesting properties. And so these are very, very massive directed evolution screens. And we did them all in mammalian cells too, because we didn't want to optimize the genes for better working in, let's say, bacteria. We want them to work in neurons. And so the pipeline you can see on the left, and on the right you can see how important it is to screen along multiple quality axes at once. If you screen for localization, you can improve localization, but brightness might not get better. If you screen for brightness, you might get something brighter, but it might not be better localized. And if you screen for both, you can optimize along both axes at once. So this is kind of a crowded slide, but let me just summarize a few key points. In the upper left, we get really good localization compared to the parent, quasar 2. It's somewhat brighter as well. In the lower right, we've improved the photostability. In the lower left, it's still quite fast. Quasar 2 is a very fast molecule. And in the upper right up here, it has good change in fluorescence or dynamic range and good signal to noise. So what that means is, this is from Bernardo Sabatini's group, you can use this molecule that we call Archon-1 where you shine red light on it and it fluoresces with far red light, 660 nanometers and redder, and you can get, for example, the monitoring of synaptic events. So here we're stimulating, or they're stimulating, I should say, in layer 5-6 of the mouse cortex, and patching and imaging layer 2-3. And in the lower left-hand corner, you can see in black the patch clamp synaptic events that are measured, and in magenta is what you see on a regular old one-photon microscope image without averaging. It works in other species. Since we're almost out of time, this just shows some data from larval zebrafish, where again you can see spiking events and synaptic events shown in magenta where my mouse is pointing. C. elegans, working with Steve Flavell here at MIT, we could also see synaptic events between two defined neurons. So can we monitor the connections between cells in real time during behavior? And most recently, collaborating with Shuo Han's group at BU, we've been working to get population voltage imaging working in awake behaving mice. So these are mice that are head fixed below a one-photon microscope. This is work spearheaded by Kirill again, but also Waan Seng, Seth Ben-Susan, and Sanaya Shroff from Shuo's group. And you can see here traces that look like electrode traces that are being imaged on the microscope from the cortex, from the striatum, and from the hippocampus. And we can see populations of neurons with this method. I think the previous record that was published so far is to image the voltage of 2 individual neurons at once. Here you can see that we image 8 in the awake mouse hippocampus. The traces are shown on the right. And now we are routinely getting up to a dozen or even more neurons per field of view. And our hope is that this will get better and better as we refine our optics and so forth. Finally, because it's a red light emitting molecule, we can use it in conjunction with blue light optogenetics and be able to activate neurons at the same time as imaging their voltage. So these are 3 neurons, again, awake mouse hippocampus. The blue bars where my mouse is pointing is when we activate, and you can see the 3 neurons here imaging the voltage while we control them at the same time. So to summarize, we've been working on optogenetic tools for controlling neurons with light, and more recently, to improve our luck, we've been evolving molecules that can report on voltage. And in the years to come, I'm really excited about combining these tools. Why can't we image the high-speed dynamics of the brain using new indicators and new microscopes, control the activity using optogenetic control, and then at the end of the day, get molecular and wiring maps so that we can combine these data sets into interesting models of how neural circuits compute. So very excited to see this hopefully take shape over the next decade, at least in small brains. Hopefully with technology help us scale up to larger brains quite soon. I think I'll end there. And I've mentioned all the people who've led projects, I believe, but let me just end on this slide, which includes many of the people who collaborated with us and have worked in the lab to make these projects reality. Thank you so much.
Thank you so much, Ed. And I see questions coming in already.
Ed, absolutely beautiful work. My quick question, okay, maybe not a quick question. My question is about the discrepancy between the extremely high resolution interventions that you're able to do and these absolutely stunning diagrams that we see of, thousands of neurons connecting to each other. My question is, how close are we to combining these two types of technologies? In other words, can we somehow learn the wiring to sufficient resolution that we can say, now I want to signal this neuron, get its output maybe through light or something to this next one, and then figure out how our more complex signals somehow transmitted through multiple neurons through the brain?
Right. So to do that, we have to link the dynamics and the anatomical pictures together. And so that's actually one of the areas now that we're working on. Can we do the imaging of a given organism and then get the wiring and molecular composition of the very same organism? And that requires engineering as well as software in order to connect those data sets. So that's still very much a work in progress. So we'll check in a year. Hopefully.
Time for one more question.
So very beautiful work. So in terms of your expansion, is there a physical limit, like how many times you can expand?
So if we polymerize and expand and form a second polymer and expand again, and then we, especially if we break the first polymer, which will eventually resist, we have done proof of concept expansions that are 3 in a row. And these result in enormous expansions. But we have not validated the resolution of that yet. It's more of just a demo that we can do it.
Okay. Thank you so much, Ed.